Hello, my name is Regina Bardi. This video is made for the MCDB 427 course, also called Molecular Biology at the University of Michigan. Today, we will be talking about an experiment that explores the binding of the Tatabox binding protein of TF2D to the Tatabox. This experiment was performed by researchers Starr and Hawley and can be found in Figure 11.5 of the Molecular Biology book, 5th edition, written by Weaver. Before I explain the details of the experiment, I want to give a little information about what the scientists knew before they began. First, they knew that a complex of many factors forms at the promoter of the DNA before RNA polymerase II begins to transcribe. One of the factors that binds to the Tatabox of the DNA early in the complex is called TF2D, or D as shown in blue in the figure. Second, the scientists knew that TF2D is a multi-subunit factor that is composed of 13 Tata-binding protein-associated factors, called TAFs, which are the numbered components shown in the figure. TF2D is also composed of a Tatabox binding protein, shown in a dark blue color in the figure. It is a conserved protein among eukaryotes, and as its name reveals, it binds to the Tatabox of the DNA. In this experiment, we will be studying the Tatabox binding protein. Third, they had an idea of how DNA-binding proteins directly contact the nucleotides. Specifically, they bind to double-stranded DNA, and one way that they interact is through hydrogen bonding. The side chains of the amino acids of the proteins form hydrogen bonds with the hydrogen bond donors and the hydrogen bond acceptors of the nucleotides. The figure shows the amino acid glutamine interacting with the thymine-adenine base pair. Although it is not shown in the figure, a protein reads a set of specific nucleic acids in order to bind, not just one base pair. Proteins bind to either the major groove or the minor groove of the DNA, since the major groove has different information than the minor groove. First, we will look at an adenine-thymine base pair as an example to show this. The top face, shown in red, represents the view from the major groove, while the bottom face, shown in green, represents the view from the minor groove. As you can see from the perspective of the major groove, this base pair has an acceptor, a donor, and an acceptor, while from the perspective of the minor groove it has two acceptors. Similarly, the major groove of a guanine-cytosine pair also looks different from its minor groove. The base pair has an acceptor, an acceptor, and a donor in the major groove, while it has an acceptor, a donor, and an acceptor in the minor groove. Looking at a base pair from the minor groove or the major groove is a similar idea to looking at a coin from two different sides. Each side has a different impression. In addition, the major groove has more information than the minor groove, since both thymine and cytosine have an acceptor, an oxygen, in the minor groove, shown here. They look identical, and the difference between the two bases cannot be distinguished from the minor groove. They can be distinguished, however, from the major groove, since thymine has an acceptor, while cytosine has a donor. Most proteins usually bind to the major groove. Starr and Hawley proposed a hypothesis that the Tatabox binding protein binds to the minor groove. They decided to test this hypothesis by changing all the bases of the Tatabox in such a way that the surface of the major groove of those bases changes, but the minor groove surface remains identical. They accomplished this goal by substituting the base thymine with the base cytosine and the base adenine with the base inosine. This was possible because, as you can see in this figure, and as mentioned earlier, in the minor groove both thymine and cytosine have an oxygen, which is an acceptor, and thus they look identical. They have different patterns in the major groove, however, thymine has an acceptor, while cytosine has a donor. The same idea applies to adenine and inosine. They both have an acceptor, a nitrogen, in the minor groove, so they look identical from the minor groove, while they look different from the major groove. Adenine has a donor and an acceptor, while inosine has an acceptor and an acceptor. Thus, the Tatabox sequence TATAAAA was changed to the new sequence CICIIII. We will call this the CICI box. In the CICI box, cytosine will base pair with inosine. The idea was to use a gel shift assay to check if the Tatabox binding protein can bind to the CICI box. Before we go on, I want to talk a little bit about how gel shift assay works. A gel shift assay is usually used to test if a protein can bind to a DNA sequence. This assay is based on the idea that a DNA fragment bound to a protein will have a slower mobility than when it is not bound to a protein. First, the DNA is labeled radioactively usually. It does not matter if it is labeled on the 5' end like it is shown here or on the 3' end as long as it is labeled. Next, the DNA is incubated with the protein for some time so that the protein is allowed to bind to the DNA and form a complex with it. Then, the complexes are loaded onto a gel and are subjected to gel electrophoresis. In this experiment, they use a native polyacrylamide gel. Then, the gel is autoradiographed to detect the DNA fragments. The DNA fragments with a faster mobility move toward the bottom of the gel, as shown here. Lane 1 in the figure contains DNA that is not bound to a protein, also called bare DNA in the figure. In this experiment, we will call it input DNA. As you can see, the DNA has fast mobility. Lane 2 contains the same DNA, but it is bound to a protein. As you can see, a shift has occurred, because when a protein binds to a DNA, it retards its mobility because it creates a complex that is bigger than the DNA by itself. For the method of the experiment, they used 3 different DNA oligonucleotides. First, they used a radio-labeled double-stranded Tata oligonucleotide, which is DNA that contains the Tata box, shown in red in the figure. This was necessary in order to compare how the Tata box binding protein binds to the Tata box versus how it binds to the CI-CI box. In test tube A, they incubated the oligonucleotide with a yeast, Tata box binding protein, and buffer for 60 minutes to allow a complex to form. In test tube B, they used the same DNA, but used a human Tata binding protein instead. In test tube C, they incubated DNA with just buffer and no Tata box binding protein. The reasoning behind test tube C is to use it as a control to see where the input DNA will be located on the gel. They repeated the same steps but used a radio-labeled double-stranded CI-CI oligonucleotide that contains the CI-CI box shown in green in the figure. They added the yeast Tata box binding protein in test tube A, the human Tata box binding protein to test tube B, and no Tata box binding protein to test tube C. Lastly, they used a labeled double-stranded DNA oligonucleotide that does not contain a Tata box and is called nonspecific DNA, shown in blue in the figure. This was just a control to show that the Tata box binding protein needs a Tata box in order to bind. It does not just bind to random DNA. They also repeated the same steps as before. Then they performed a gel shift assay. When they ran the gel, they observed these results. The first 3 lanes contained the CI-CI oligonucleotide, the middle 3 lanes contained the Tata oligonucleotide, and the last 3 lanes contained the nonspecific DNA oligonucleotide. Lanes 1, 4, and 7 contain the yeast Tata box binding protein. Lanes 2, 5, and 8 contain the human Tata box binding protein, and lanes 3, 6, and 9 contain no Tata box binding protein. First, we will take a look at the lanes that contain the Tata box oligonucleotide. Lane 6 only contains 1 band at the bottom. We will call this input DNA. No shift is observed here, which means no Tata box protein DNA complex formed. This is what we were expecting, since that lane does not contain any Tata box binding protein at all. Lane 5 has 2 bands. The top band is a shift, and it shows that a complex must have formed since the DNA in that band has reduced mobility. There's also a bottom band there, which is the input DNA, and it shows that not all the DNA was bound to the Tata box binding protein. In lane 4, we can also see 2 bands. The bottom band with the faster mobility is the input DNA, and the top band is a shift that shows a complex formed. The complex when the yeast Tata box binding protein was used in lane 4 has faster mobility than the complex in lane 5 when the human Tata box binding protein was used. This is because the human Tata-box binding protein is bigger, so it retains mobility to a greater extent than the yeast Tata-box binding protein. Next, we will take a look at the first three lanes that contain the CI-CI oligonucleotide. In lane 3, we can see there is no shift because no protein is present. In lane 2, we can see that there is a shift, and thus can conclude that the human Tata-box binding protein was able to bind to the CI-CI oligonucleotide. The yeast Tata-box binding protein also binds to the CI-CI oligonucleotide since there's a shift in lane 1. The yeast Tata-box binding protein creates a complex with a faster mobility than the human Tata-box binding protein since it is smaller. Since both Tata-box binding proteins were able to bind to the CI-CI oligonucleotide, it means that the Tata-box binding protein was unable to tell the difference between the CI-CI oligonucleotide and the Tata oligonucleotide, so it must bind to the minor group. Lastly, we will take a look at lanes 7, 8, and 9. They contain the nonspecific DNA oligonucleotide. Lane 9 does not have a shift, since no Tata-box binding protein is present. But we can also see that lanes 7 and 8 do not have shifts either, and thus no complexes formed in these two lanes. This is accurate with what we were expecting to see. It shows that neither of the Tata-box binding proteins can bind to a DNA sequence that does not contain the Tata-box. In conclusion, since the Tata-box binding protein requires a Tata-box in lanes 7, 8, and 9, but it can still bind to the CI-CI box in lanes 1, 2, and 3, then the Tata-box binding protein binds to the minor group of the Tata-box. Thank you so much for watching.